This is Bio 1, Biology 1440, micro lecture on chemistry. This won't replace normal lecture or your textbook reading, but it's a supplement for your textbook and your reading. And we're going to talk about the basic chemistry today, so chapter 1, or chapter 2, 3, and 4 essentially. And first things first is we start with atoms. Just so we can understand what's coming later, we need to start with the beginning, and the beginning here is atoms. And the nucleus of an atom consists of subatomic particles, some of which have a positive charge, some of which really don't have a charge. The ones that have a positive charge are the protons. The ones that have no charge are the neutrons. And these are found in the nucleus of atoms. Now, the number of protons equals essentially, well, I should say the number of protons determines the element, what that atom is. Remember, an atom is the smallest unit of an element that still has the properties of that element. So you can have an atom of gold, or an atom of silver, or an atom of hydrogen. So this is the nucleus, and outside of that nucleus will be electrons, and they don't orbit like planets around a sun, but we tend to draw them at various energy levels. Electrons have orbitals that are in odd shapes, not exactly spherical, but we draw them in energy levels starting inward towards the nucleus. The first energy level can have 2 electrons in it, and every outer energy level outside of that can have up to 8 in it, the rule of octets. So in this case we have 2 protons in this atom, so we would also then have 2 electrons, and electrons have a negative charge. So the number of electrons usually will equal the number of protons, unless we have an ion, and we'll talk about that in a moment. Okay, so this leads then to the possibility of bonds, chemical bonds. So chemical reactions are going to be the breaking and reforming of bonds between atoms. So we take atoms and we stick them together to make molecules. And those molecules could be composed of all of one type of atom, like a big chunk of pure gold, or they could be composed of lots of different atoms, a compound, a molecule made of several types of atoms. So the first type of bond I'm going to mention is simply called an ionic bond. An ionic bond is going to be found in substances like salt, for example. They come apart easily, especially in water when substances dissolve. We'll talk about that soon as well. And let's just do an example over here, same thing from your book, of sodium and chlorine ions. So I'm going to draw sodium here. We have our first energy level, and it's going to have 2 electrons in it. And then we'll have another energy level out here. It's going to have 8. Okay. Then we have another, whoop, that messed up. Hold on. Another energy level out here, and it is going to only have 1. Now obviously that outer high energy level, the farther out we go from the nucleus, the more energy we have in those energy levels, and that electron isn't happy, that atom isn't happy. atoms are stable when that outer energy level has a full complement of electrons, in this case 8 electrons, and here we only have 1. Now this atom over here, chlorine, has 2, then it has another energy level with 8, has another energy level out here with 1, 2, 3, 4, 5, 6, 7. So the outer energy level here is missing 1. So if this electron leaves and comes over here, so an electron is actually lost. I'm going to abbreviate electron like this. Lost, and actually moves to the chlorine. So at this point now, if we erase the electron, actually I could erase this whole circle because there are no electrons there. We put it over here. Now this sodium is missing an electron, so it becomes more positively charged because electrons are negatively charged. So if we lose an electron, we have one more proton than we do electrons now. Now our atom is now technically an ion. It's charged, and this chlorine has gained an electron. It becomes negatively charged. And as we know with magnets, positive and negative charges are attracted to one another. And this attraction now between these two, because of their charges, is an ionic bond. It's a bond between ions because of their charges. Okay. We also have hydrogen bonding as a possible type of bond. And let's just draw a water molecule here. Here's water with, it's got oxygen here, and these out here are hydrogens, H and H. Okay. This is a type of covalent bond that we'll mention in a moment, where electrons are not shared equally. So oxygen has a much stronger affinity for electrons than hydrogen does, so the electrons spend more time over here. And actually, let's just talk about covalent bonds while we're at it. So let me draw a bigger water molecule here. Looks like Mickey Mouse's head. Alright, this is oxygen. This is hydrogen. Here's another hydrogen. And they're sharing electrons. So when you share electrons, rather than steal them like we saw in ionic bonds, this is sharing. And ionic bonds are somewhat weak. This is a very strong bond. Electrons are being shared here. Hydrogen has 1 electron. Normally, its first energy level can hold up to 2, and it shares an electron with hydrogen or with oxygen, and it therefore completes that energy level. That first energy level now has 2 electrons, although one of them is borrowed. Now, oxygen borrows electrons more frequently from hydrogen than it lends electrons to hydrogen. Therefore, it has a polar covalent bond. Now, if you have a carbon stuck to a hydrogen, they share electrons equally, and that is a nonpolar covalent bond. You can read about this in your textbook if you are confused. Oops, there we go. Which means equal sharing of electrons. So they both have equal electronegativity, so one does not borrow the electrons more than the other. They share equally in carbon-hydrogen bonds. But in oxygen-hydrogen bonds, oxygen is more electronegative. It gets the electrons more frequently, and therefore, oops, I have a hard time drawing the sigma sigma shape, but it is more negatively charged than the hydrogen, which is going to be more positively charged. So if you look in your book, you'll have kind of an S shape, sort of like this, sigma, sigma, sigma plus, sigma minus, which just means that this hydrogen is more relatively positively charged than this oxygen, because the oxygen has the electrons more often. Electrons are negative, so if I'm holding on to the negative thing more than you are, I will be more negatively charged than you. Okay, so covalent bonds hold all kinds of matter together. They're very strong, and sometimes they result in charged molecules, like this water molecule having a positive end and a negative end. So this is a polar molecule. So these are covalent bonds holding hydrogen to oxygen. But hydrogen bonds now, because this is somewhat, we'll do the sigma plus on this end, and this end down here is somewhat negative. Now another water molecule over here might orient this way. Remember, this is positive, this is negative. They will form a bond, because kind of like in ionic bonding, positives and negatives attract. Right here is a hydrogen bond. So hydrogen bonds are essentially an emergent property seen in this case in water, which occur because of the unequal sharing of electrons between oxygen and hydrogen in the atoms that are comprising that water molecule. So the oxygen side has a negative charge, hydrogen side has a positive charge, therefore they can interact with neighboring molecules, and that is the hydrogen bond. So bonds between water molecules are hydrogen bonds. Bonds in a water molecule are going to be covalent bonds, in this case polar covalent bonds. And these hydrogen bonds give water tons of cool properties that make it useful for life on Earth. Okay. For example, some of the properties of water, and you can read about these. Properties of water are going to be high specific heat. Oops, that looks horrible. Sorry about that. High specific heat, which means it takes a lot of energy to warm up water, and it takes a lot of loss of energy to cool off water. This is why having an ocean nearby stabilizes your climate quite a bit. So this basically is how much specific heat is measured is how much heat energy it takes to change 1 gram of water by 1 degree Celsius. So how much energy does it take to heat up 1 gram of water by 1 degree Celsius? And it's measured in calories, and that's where our unit of calories comes from. Okay. It also has a high heat of evaporation, meaning because these water molecules stick together, it's hard to get one to break away and evaporate, so it takes a lot of energy to do that. This is really useful because this means when water evaporates, it cools off the surroundings. So this is why when you sweat, you cool off. You put a bunch of heat energy into that water, it breaks away and carries some of that energy away, thereby cooling you down. Same thing with the ocean. The oceans evaporate, helps to cool off that part of the earth. And also, because of hydrogen bonding, solid water is less dense than the liquid. And you can see why in your textbook. When water stabilizes as a solid form, the hydrogen bonds keep all the water molecules at a distance from one another, which makes it less dense. And because of this, ice floats and acts as an insulator to the water below, which helps life to survive winter. If I sank, then there would be no life on Earth as we know it, because fish and other marine organisms would probably be crushed or frozen under blocks of ice that sink to the bottom. Okay, let me turn that off and go to another page here. Okay, so another thing I want to mention excuse me, is that water, because of its polarity, its charge, it's a really good solvent. So if you have a sodium ion, Na+, or chloride ion in the water, it can dissolve. Water is a solvent, which means it can dissolve solutes, creating a solution. So a solvent is the liquid, and you add to that a solute, which is the solid dissolved in there, and this all equals a solution, which you'll be making a lot of in lab. Okay, so water, because of its polarity, here's a positive molecule. Now, water has a positive end and a negative end. Which end will be attracted to this sodium? As you probably know, opposites attract. So, here we go. The negative end of the water will face the sodium, and this will happen all around this sodium. So we're forming essentially like a bubble of water around this sodium ion, and the opposite will occur here, where the positive hydrogens of water will face, well, that's kind of messed up, oh well, will face the chloride ion. These are called hydration shells, and this allows for things to dissolve in water. So water essentially is stuck to the sodium or the chlorine. They're stuck to it through hydrogen, well, through I guess you could say through hydrogen bonds, and that allows that substance to be dissolved. It's now floating around freely in the water. It's actually stuck to the water directly, and that's what it means when things dissolve. Another cool thing about water is pH. Water disassociates, meaning that if you have two water molecules, every once in a while, though rare, they come apart and come back together again, yielding, in this case, OH and hydronium. And when you have a bunch of this, which is essentially, you can think of this as just a free hydrogen ion, but it actually tends to stick to another water molecule, forming hydronium. When you have a bunch of this, when you have more hydrogen ions or hydronium in a glass of water than you do OHs, then you have an acidic situation. When you have more OH than you have H, then you're going to have more of a basic situation. So water tends to naturally disassociate, and we measure the concentration of hydrogen ions, but in reality we think of it as hydrogen ions, but just remember, in reality they're actually stuck to another water molecule forming H3O. They're not actually just floating around naked. The H pluses stick and form H3O, hydronium. But when we measure those hydrogen ions, so the concentration of hydrogen ions in pure water would be, if you were to measure them, would be 10 to the negative 7 moles per liter. And if you don't remember what a mole is, look it up. and if we take take this negative 7, turn it to a positive, and just say pH of 7 is neutral water. It comes from the measurement of how many hydrogen ions are in that water, or how many hydronium ions are in that water. So pH of 7. So we're measuring the hydrogen concentration here. So another example, and basically this pH scale is a logarithmic scale. So every change in pH, so if you go from a pH of 5 to a pH of 4, that's a 10 times difference in the concentration of hydrogen ions. And we take the negative logarithm of the concentration of hydrogen, and that's how we get our pH. So we go 10 to the negative 7. What's the negative logarithm? That's 7. All right. So if we had a pH of 5, so that would be a hydrogen ion concentration of 1 times 10 to the negative 5. If we had a pH of 4, that'd be 1 times 10 to the negative 4, and that would also be the concentration of hydrogen ions. So 10 to the negative 5 is fewer hydrogen ions than 10 to the negative 4. It's a 10 times difference. And if you're curious, how do you figure out what the concentration of OH is in this case, hydroxide? Just take, so let's say this is a pH of 5. Figure out what plus 5 equals 14, and that's how you know what the concentration of OH is. So let's say in a pH of 4, OH concentration, oh well, is going to be 1 times 10 to the negative 10, which is a very small number. In this case, it'd be 1 times 10 to the negative 9, okay? Which is a slightly higher number. So a little bit more basic in that case with a pH of 5. So pH of 5 is a higher pH than a pH of 4. And the lower your pH, the more acidic you are, the more hydrogen is floating around. So the more hydrogen you have, so the greater the hydrogen equals more acidic. So a low pH means more acidic. So spend some time and work that out. Another thing about pH, and especially in humans and mammals and all organisms with pH systems in their blood, is we can regulate pH in our blood. It's very important to maintain internal conditions, the process of homeostasis. And we do this through blood buffers. So a buffer is going to resist changes in pH. And we have a buffer in our blood when we breathe. Through the process of cellular respiration, we take in oxygen and we break down food and we release carbon dioxide as a waste product, which makes its way into our blood. Our blood has a lot of water in it. That's going to yield H2CO3. And of course, this process is reversible. This is going to be carbonic acid, which is going to be a weak acid. Now, right now, it hasn't changed the pH of our blood because it hasn't dissociated yet, but it can dissociate now into bicarbonate and hydrogen. And it can go back the other way. So if your blood becomes very basic, it's becoming more basic, the carbon dioxide that's all over the place in your blood can form this acid, dissociate, and increase the hydrogen ion concentration. If your blood becomes too acidic, the hydrogen ions can stick to the bicarbonate in your blood, and we can actually then remove it from the blood, and eventually this CO2 is going to leave the lungs and be breathed out. So we can maintain pH using carbonic acid. It's a weak acid that dissociates into essentially forming a weak acid and a weak base. And once again, if hydrogen ions are too high in your blood, we can remove them from the blood and raise the pH. If your blood is too basic, then we can form more carbonic acid and release more hydrogen ions, and we can lower the pH. And you can see that in this reaction and read about it more in your book. Okay. Now, I think we ought to do a quick summary of the carbon chapters on organic chemistry. And carbon is the building block of all living things that we know of, and it has an energy shell with 2 electrons and an outer energy shell with 4 electrons. 1, 2, 3, 4, and of course that means it can still stick 4 more on there. This makes carbon very reactive, essentially. It can stick to lots of different things. It can form complex molecules because it has 4 sticky areas. Each of those electrons is naked, does not have a dancing partner. Each one wants a partner, meaning it can form lots of bonds, either with more carbon or with other molecules. So an example of like a hydrocarbon, which is just carbon hooked to hydrogens, would be something like this. Okay, so a basic hydrocarbon. Now what we're going to do for life is we're going to stick functional groups. So we will have large carbon-hydrogen backbones, but we're going to stick functional groups onto those backbones that give each of those molecules special properties. And also carbon can form rings, it can form different structures, it can form a complex variety of molecules, making it great as a building block for living things. And we can change the properties of these molecules by adding different functional groups. So we can change the molecule. Now this molecule here, lots of carbon and hydrogen, all of these are nonpolar covalent bonds, which means this is a molecule that does not mix well with water because water is polar. This molecule is not polar. So it's nonpolar, or as we would say, hydrophobic. However, you can have organic molecules that mix with water because they will have polar functional groups that make the overall molecule polar. Now, the major types of organic molecules that we're going to concern ourselves with are carbohydrates, and carbohydrates such as glucose, sugars, are used for energy storage. They're going to be used for cell-to-cell communication, as we'll see later, but another major role of them is going to be structure. You might not think of them as being important for structure, but they are. So glucose is the carbohydrate we're going to focus on most in this course. It's C6H12O6, and it forms a linear molecule, but when it dissolves in water it forms a ring. You probably know how to number the carbons on the ring. So this is one molecule of glucose. This is a monosaccharide. Monosaccharides are single rings, and of course not all of them are 6 carbon. So there are 5 carbon sugars as well, but I'm just going to use glucose as our model. We can also take a couple of rings and stick them together through covalent bonds and form, kind of looks funny, but that's okay, disaccharides. And of course we can have a bunch of them stuck together. I'm just going to draw them as circles for time's sake here. Lots of them stuck together, and that'd be a polysaccharide. And these are stuck together with covalent bonds through dehydration reactions, and these are technically glycosidic linkages, or glycosidic bonds. So we have a polymer here. This is a polymer composed of the monomer glucose. And these are going to be used, as I said, for energy storage, for energy use. Our bodies take glucose and run it through aerobic and or anaerobic respiration. Also, plants stick a bunch of glucose molecules together to make a polymer called cellulose. Insects and fungi stick it together to make a molecule called chitin. Both of these are very hard to digest because the bonds between the monomers are in such a shape that our enzymes cannot get in there and break those bonds. So structure and function come together, and the way that those bonds are put together, you can read about it more in your book, but these bonds are hard to break in that case. However, other types of molecules like starch, which are also composed of a bunch of glucoses stuck together, but they're, as you'll read in your book, slightly different isomers here. We can end up with bonds that are much easier to break in this case. Now, we have, once again, monomers, single units. So this is a monosaccharide, disaccharides such as table sugar, polysaccharides comprise the different types of molecules that we'll see when it comes to the carbohydrates. Now, lipids are another type of molecule we want to talk about, and we're going to talk about fats especially. These have a lot of carbon to hydrogen bonds, which make them nonpolar, which means they don't mix well with water. And if we have a fat, we'll see that, I'm going to draw this, I should draw this sideways, but see 3 carbons stuck together here. This is a glycerol. And there's going to be 3 fatty acid tails stuck to that. So this is a fat molecule. This is great for energy storage. Fats are also used, or lipids are also used for communication in the form of hormones, but obviously lots of energy storage. In fact, twice as much energy storage here as you would find in a carbohydrate, gram for gram. If we were to zoom in on a fatty acid tail, we would see lots of carbon to carbon and hydrogen bonds here, okay? Now, those bonds between carbon can be saturated or can be double bonds or single bonds. And when you have double bonds, you're using more electrons to hold the carbon to the carbon, which means you can hold fewer hydrogen ions. So, or hydrogen atoms. So if you look at this middle carbon, it's saturated. It can only hold 2 hydrogens, and it's holding 2. It's a saturated fat. Now, if you had a tail that had this going on, we would then be able to hold fewer hydrogens. And this would actually cause this fatty acid tail, rather than being straight, it would cause it to kink. And remember, structure and function are related. Kinked up tails mean your molecules can't pack as closely together, which means at room temperature your unsaturated fats are going to be more like oil or vegetable oil is a good example. It's going to be liquid, and that changes the property of the fat. Whereas saturated fats, they have straight tails, they can compact very closely together, and they will be solid at room temperature, and that will affect their function in the human body. Another type of lipid I want to mention is just phospholipids. I'm going to draw these very quickly. They have a phosphate group on one end and 2 fatty acid tails. They could be saturated or unsaturated tails, and these are going to form our cell membranes that we'll see in future chapters. They can orient themselves like this and make a lipid bilayer. They have a polar end and a nonpolar end. So this end here is polar. actually it's not positive, but it's polar, and this is nonpolar. Okay, and we'll see that later. They make up cell membranes. All right, now proteins. Proteins are composed of a monomer called amino acids. Now, the lipids we saw above are not a monomer-polymer relationship, but proteins, carbohydrates, and nucleic acids do have monomers that come together to build a larger polymer molecule. And now, amino acids consist of a central carbon, we'll have a carboxyl group on one end. This is an H here. And then we'll have an amino group on the other end, which has a nitrogen in it. This is why nitrogen, one of the reasons nitrogen is so important for life on Earth. And we're going to have an R group, which is basically just a this box represents any of the functional groups that could be there that will alter the properties of this amino acid. So the part we have drawn down here is just the backbone. It's the R group that changes from amino acid to amino acid. So that's the one we need to pay attention to the most. Now that R group can have different properties, which will make the amino acid either polar or nonpolar, or I should say hydrophilic or hydrophobic. So if that R group has a lot of carbons stuck to hydrogens, and like that, or not really like that, it'd probably be more like this, like we've seen before, etc., etc., etc. This would be a non-polar amino acid. Now, if there are carbons stuck to carbons and there's an oxygen stuck somewhere on there, or there's a carbon stuck to a carbon and there's a nitrogen and it has a big plus sign next to it, you would know, okay, oxygen, very electronegative. This is probably a charged molecule. It will react well with water. It will associate with water. If there's a positive or a negative sign on it, that molecule will also react well with the polar water molecule. But if it's a non-polar bond, so if it's like this, or a lot of the R groups that are in rings, those are often non-polar as well. You can read about this more in your book. But the changes in the R group change the properties of the amino acid. So be able to recognize this and be able to tell by looking at an amino acid. You don't have to memorize the amino acids, but learn how to tell the difference between those that would and would not react well with water. It's very important to understand that, because when these are all stuck together, you have one amino acid stuck to another, 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 etc., etc., etc. This is how we build a protein. And proteins rely on their shape, their structure influences their function. And the properties of the R groups allow this protein to have very specific shapes. So we have all of these amino acids stuck together with covalent bonds called peptide bonds. And these are different amino acids. Some of them are polar, some of them are nonpolar. So if this were in water, this whole thing might fold up, and the nonpolar amino acids would go to the middle, and the polar amino acids would go to the outside and interact well with the water. So that will help to shape the amino acid. So the order of amino acids is called the primary structure of a protein. That's just the order of the amino acids, just like reading a sentence, which letter comes first, okay? Secondary structure occurs then when these interact, and we'll talk about this more in lecture, but they start to fold either into a pleated sheet or a helix, which I can't really draw, but I'll just draw a curlicue here. So this is going to be your secondary structure. So these are basically going to be interactions between the backbones and things on this primary structure holding together to make these shapes. And then we're going to have tertiary structure, which is when R groups, like an R group over here is attracted to an R group over here, and it folds this whole thing around, creating a glob. So this is how we get globular proteins. And then if you get more than one tertiary structure, and you stick them together. So here's a globular protein and it sticks to another globular protein and maybe another globular protein. And this is how we get complex proteins with quaternary structure. And these will have very specific shapes and will allow us to have enzymes and structural proteins that all have a specific job, whose shape is very, very important for that job to be done correctly in the cell. So have a look, read over the structures of proteins, what kind of bonds hold them together, and how they work. Lastly, I want to just mention nucleic acids. Nucleic acids are energy storage in the body. So we're going to have RNA and DNA. Ribonucleic acid and deoxyribonucleic acid. And they're both composed of nucleotides. That's the monomer, the unit that comes together to form nucleic acid. So nucleotides are going to consist of a central sugar. See if I can, let me try that again. It's going to be a 5-carbon sugar. It's going to be attached to a phosphate group, which has a negative charge. And it's going to have some type of nitrogenous base. Here. Twink. And then down here, it's going to have several things attached to these carbons, but right here, if you look and see whether it has an H or an OH, so I'll put an OH on this one. If instead, well, let me just put it this way. If it were an OH, this would be a molecule of a monomer of RNA. If it were an H, it would be a monomer of DNA. So have a look. And when you look at the book or read the book, it'll show you this. You can tell whether your molecule is DNA or RNA just by looking at the nucleotide, whether there's an H or an OH at this position. Deoxyribonucleic acid, or DNA. Deoxy means it's missing an oxygen. Okay, now this nitrogenous base can be of two types. It can be a single ring. I'm just going to draw a very crude ring here, and this is a pyrimidine. Or it can be, let me just draw a crude ring here. It can be a couple of rings stuck together. A double ring, which is a purine. We'll talk more about those later on in the book. But what happens in DNA, let's just focus on DNA for now. Your sugar and phosphate groups here make a backbone. So you'll have your sugar and your phosphate group, and it will stick to another sugar and a phosphate group. And a phosphate group. And then down here's a sugar and a phosphate group. This is the backbone, and there'll be another one over here. So these are the backbones of the DNA. And these nitrogenous bases stick out, and purines and pyrimidines always stick together. So we'll have these bases sticking out, forming hydrogen bonds, holding your double helix together. And whenever this happens, there's always a purine on one side, and it's bound by hydrogen bonds to a pyrimidine on the other side. So they always pair up that way. And this is what allows us to code for genetic information. And this is how we form then the central dogma of molecular biology, which is that DNA is a code. It codes for RNA, which is a messenger, that eventually and it's a blueprint for the eventual production of proteins. And this is what DNA does. DNA codes for the primary structure of proteins, And the primary structure of proteins then folds up to make the secondary structure, which folds up to make the tertiary structure. And not all proteins have a quaternary structure, but some do. But either way, the protein shape then allows it to function. Without the proper shape of DNA, or the proper sequence of DNA, you don't get the proper protein. You have a mutation, and that changes the shape of your protein, which results in usually a malfunction in the organism. Alright, that was a whirlwind run through. I didn't go into tons of detail there, but I just wanted to do a summary. Some of the stuff you'll have to read through in the book to flesh out the details, but I hope this helps. See ya.